Hello, and thank you for joining us. I'm Gwen Taylor, Senior Editor with Current Protocols at John Wiley & Sons, and I'm here today to introduce the webinar entitled Multiplexed Protein Detection Assay Fundamentals and Applications in Cancer Research. This webinar is being co-sponsored by Current Protocols and EMD Millipore. EMD Millipore is the life science division of Merck KGaA of Germany and delivers products and services for life science research in academic and pharmaceutical environments. They offer the largest portfolio of biomarker assays, including ELISAs, radioimmunoassays, and multiplex assays that enable researchers to study complex biological systems and identify new diagnostic and therapeutic targets. EMD Millipore is a preferred distributor of the entire Luminex portfolio of multiplexing instruments, and their multiplexing panels are manufactured to industry gold standards, giving users the same accuracy and precision in every lot, backed by highly knowledgeable technical support scientists. Current Protocols is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 15,000 protocols, Current Protocols is part of Wiley Publishers. We've allotted 1 hour for today's program, but our speakers have agreed to stay on a bit longer to answer as many of your questions as possible. You can submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. The webinar will be recorded and available for viewing in the next few days. We will send each of you an email with details on how to access the recorded webinar, along with a PDF of the slides. And now it is my pleasure to introduce today's speakers. Dr. Brian Haab is an associate professor in the Center for Translational Medicine and is head of the Laboratory of Cancer Immunodiagnostics at the Van Andel Research Institute in Grand Rapids, Michigan. His research focuses on the development of biomarkers for pancreatic diseases with an emphasis on the glycobiology of pancreas. The Haab group has developed several methods using antibody microarrays and other microarray formats in biomarker research, and has identified several candidate biomarkers that are currently being validated for the diagnosis and prognosis of pancreatic diseases. Dr. Beatriz Carreno is a research associate professor in the Departments of Medicine and Pathology and Immunology at the Washington University School of Medicine in St. Louis, Missouri. Her research focuses on tumor-specific immunity and tumor microenvironment, especially in melanoma and breast cancer. Dr. Debra MacIvor earned her PhD in molecular genetics from Washington University in St. Louis and has been at EMD Millipore for 6.5 years as the product manager of the immunology and cancer MILLIPLEX MAP portfolios. So let's begin with a very warm welcome to you, Dr. Haab.
Thank you, Gwen. It is a real pleasure to be participating in this webinar, and I'd like to thank Current Protocols and Millipore for hosting it and for inviting me to be part of it. So I'm going to talk today about multiplexed antibody assays, and I'm going to focus on practical considerations really for the research laboratory setting, considerations that you will need to think about to make this work well for you. I'm going to give an overview of the main formats of the types of assays and detection methods, and then I'm going to talk about some of the considerations for optimizing the assays and experiments for analyzing clinical samples. So first of all, why do we use antibodies and immunoassays? Immunoassays or antibody-based assays really are the workhorse for the analysis of proteins in biological samples. Many other types of assays have been developed, particularly in the mass spectrometry area for analyzing proteins, but antibodies and immunoassays remain the method of choice for specific and sensitive detection in biological samples. So they're another valuable aspect of immunoassays is that they can be used in many different platforms and formats, from microchip or bead-based, as well as microfluidic platforms. Other types of novel platforms are amenable to using antibodies. Also, the value of antibody-based assays is that they are particularly well suited for clinical studies where there's a requirement for being able to run many samples while maintaining good precision so that we can build up statistics over large patient cohorts while getting good measurements on each patient. And some high throughput assays are not very precise, and then other assays can give good multiplexing but not good precision or can't give good throughput. So antibody-based platforms have those combined capabilities. And the topic of today's seminar is multiplexing, which is the ability to run multiple immunoassays in a single run or in a single experiment. And this ability increases the value because we can get measurements on many different analytes at the same time. So I'm going to talk now about the two main types of formats that are used for multiplexing assays, and those are the planar and bead arrays. In a lot of ways they're very similar. I'm going to focus on the planar arrays because that's the format used in my laboratory, although we have some familiarity with the bead arrays. So both of them have the ability to multiplex. They use a different principle for the multiplexing. The planar assays have different capture antibodies on different locations on the array by which their different assays are distinguished. The bead arrays use different colors of beads, and toward the end of my section I will describe how the bead arrays work. I'm going to, just as the examples for what I want to talk about, the examples will be drawn from the planar arrays, but all of the considerations that I'm going to talk about would apply to both platforms. So they have similar experimental considerations and capabilities, although the instrumentation and analysis methods are different, and I think you'll hear a little bit about that from the next speaker. With both methods, there have been two main detection methods that have been used in the multiplexing assays, and I refer to those as label-based and sandwich. By label-based, I mean methods where a dye is attached to all the proteins in a sample. So we take a complex biological sample, react to fluorescent dye with all the proteins so that they all are labeled, and we use a capture antibody immobilized on the surface to capture the target that's labeled, and we can scan for fluorescence. One of the advantages of that method is that we can use a two-color format to compare a sample to a standard reference sample, which gives us good reproducibility precision between the assays. Also, because there's just one antibody per target, the multiplexing is easy. We just add multiple capture antibodies. However, the specificity is lower because we only use one antibody instead of two. In the sandwich assay, backgrounds can be high because all protein targets are labeled. The other method is sandwich assays, which is the standard method used in ELISA formats where we have paired antibodies against a single target. These have the potential for higher specificity and sensitivity relative to the label-based assays, but multiplexing is more difficult because we need a different detection antibody for every target, and there's more opportunities for cross-reactivity between the targets, and that has to be accounted for in a rigorous fashion. The commercial developers of the panels of multiplexed assays have worked through all those issues of the cross-reactivity, so that takes out that level of preparatory work for the researcher when using some of the commercial panels. But if you are developing your own panels, that needs to be systematically addressed, as whether there's any cross-reactivity between the detection antibodies. It was really for that reason that we worked on a couple of alternate formats for multiplexing I'll tell you about in the next slide. I wanted to also show you that the multiplex detection can be used for not only protein detection but glycan detection. That's a format we've really worked hard at, and we found a lot of value in biomarker studies because in many cases in disease situations, not only the protein abundance can change, but also the glycosylation on the protein can change. So we found that if we detect both the protein abundance and the glycosylation, or specific glycoforms of a protein, we can get more information about a disease state. And that's done in panel A. As you can see, it's very similar in its beginning to a standard assay. We spot down multiple antibodies, we capture proteins, but then instead of detecting the core protein, we use a lectin to detect the glycans on the captured proteins. And we can use a variety of different lectins, which are glycan-binding proteins, to detect different types of glycan structures depending on what it is we want to detect. In the lower panel, you see the standard protein detection format where we use paired antibodies to detect the core protein. So as I mentioned, we have an alternate approach to multiplexing that makes it a little bit easier for developing multiplex sandwich assays. And here we see two alternate approaches. I mentioned how when we use cocktails of detection antibodies in multiplex sandwich assays, there's high information content. We have a different detection antibody for each target, and the requirement is that each detection antibody goes to its target without any cross-reactivity. However, when we use many different detection antibodies, the opportunities for cross-reactivity increase, and many have found a limit of a few dozen assays in one single pot, so to speak, before that becomes very hard to control for. So the controlling for cross-reactivity becomes increasingly hard with increasing multiplexing. So we've decided to split up our assays into increasingly smaller assays where we use smaller and smaller numbers of different assays, but the volume correspondingly decreases. So when we reduce it down to maybe just a single assay per well, we don't have any concerns about cross-reactivity, but we use tiny wells so that when we use many different assays, we still haven't consumed a high volume of our clinical sample. And the requirement to make that really work in practice for multiplexing and for running many samples is the ability to have high throughput sample processing. I'm going to tell you just briefly about a practical method we have for running low volume, high throughput assays. And in this format that we developed in our lab, we take a microscope slide and partition it into many small arrays. We can make the arrays very small or medium sized, and each partition has its own antibody array, anywhere from just a few antibodies to many, depending on the size of the partition. And then each well would get a different sample or maybe the same sample with a different detection antibody. The way that we partition the slides is using what is called a slide imprinter from the gel company. A microscope slide is inserted upside down into the holder on top. It's a hot plate with a wax bath, and hydrophobic wax is melted in the hot plate. A stamp residing in the hot bath is elevated to contact the slide when the lever is pulled, thus imprinting a wax pattern onto the slide, which forms hydrophobic barriers. And so then a small volume of sample can be loaded into each well, and that's amenable to high throughput sample processing. We can vary the sizes of the wells, so there will be a trade-off between the size of the number of different antibodies that go in each well versus the volume that goes in each well. For example, if we print 12 arrays on one slide, we can fit about 484 spots in each array, and it takes about 40 microliters per array. A medium size that we find really practical is 48 arrays per slide, up to 144 spots with only 7 microliters per array, or if we want to use really small assays, we can do 192 with up to just 9 spots per array, but a very small volume per array. So we like this system for prototyping and for running new multiplexed assays because it really speeds up the development time when we eliminate many possible cross-reactivities in the multiplexed assays. So I'm now going to talk about some experimental considerations, things that you should think about before running the samples and in the design of the experiment and in the analysis of the experiment. So first of all, before running your samples, there are many things that you should have in place. And for each assay, it is very valuable if you have access to more than one antibody per target to optimize the antibody sandwiches. So optimize the capture and detection pairs for each target. This can really pay off in terms of sensitivity and specificity. And I'll show you in the next slide how multiplexed assays make this kind of optimization really efficient, whereas in standard approaches, much more sample, many more experiments are needed to optimize, but with multiplex assays we can optimize the capture detection sandwich easily. In addition, it's very important to have a calibration curve for each assay. This confirms that the assay is working well. It shows where the linear response range of the assay is and is used to calibrate signals to concentrations. Next, you want to have control samples. These are experimental samples that are run with each experiment that can confirm consistency between experiments. And also, you need to determine the dilution factor that is used for the sample. So the dilution factor depends upon, first of all, it's the factor by which each sample is diluted into a sample buffer. And that can either be small or large, so maybe just a 2-fold dilution or less, or it could be a 10,000-fold dilution. And it depends upon the starting concentration of your analyte. And if you have a wide range in starting concentrations in your multiplexed assay, you may need to split up your assays into those that are highly concentrated and those that are not concentrated, so that you can match the dilution factor within those assays. And the way that we set the dilution factor is we take the control samples and we run them in preliminary experiments under several different dilutions and see how the signals match up to the calibration curve. And I'm going to show you an example of that. And as I mentioned, you may need different dilutions for different assays, depending on how concentrated the starting concentration of the analyte is. So I mentioned how multiplexed assays can really speed up optimization of capture detection pairs. Here's an example of how you can do that. This is an example of 3 different antibodies used in capture detection pairs. So with 3 antibodies, each used as the capture and each used as the detection, we have 9 different combinations. So you see on the top there a picture of 3 identical antibody arrays, each printed with antibodies 1, 2, or 3 against the same target. In this case, it was against a glycan, is the way I've depicted it. And then we incubate the proteins on the antibody array. They bind to the antibodies, and then each array gets detected with one of the antibodies. So the array 1 was detected with antibody 1, array 2 was detected with antibody 2, array 3 with antibody 3. And then, as you can see, there are 9 different capture detection combinations at the bottom. And so then we can look at each combination and see which one gives the best performance. And here's an example of an optimization for the analyte CEA, cancer marker CEA, with 4 different antibodies that were each used as the capture and detection. So each plot shows the curves with each of the antibodies as capture, and each plot is a different detection. So you see there are 16 combinations there. And what to note from here is that in each plot, the self-self pair doesn't work. These are all monoclonal antibodies, so they are against a specific epitope on the protein. And as is well known in developing sandwich assays, if you have a capture and detection targeting the same epitope or a very close region, it's not going to work well as a pair because they compete for the same place on the protein. So that matches up with what we see here, and then we can simply look at the range of concentrations and the shape of the curve as to which one works best. And from this, it looks like using antibody 3 as capture and antibody 2 as detection works best. So selecting that combination as our optimized pair, then we can use that to look at the linear response range of the assay to set our dilution factor, as I mentioned before. So on the left panel here, we see the curve over a range of CEA concentrations. We had purified protein as our standard. And when the CEA is too concentrated, you see above about 200 nanogram per milliliter, the curve starts to flatten out, to plateau. It's because we are saturating the binding of the capture antibody. And at that point, we no longer can get linear responses. And then if the CEA is too dilute, so down near 2 nanogram per milliliter or so, we can no longer pick up the signals. So in between that range, the fluorescence values from about 25,000 down to, say, 10,000 or so is our responsive range. So what we're looking for is signals in that range. So then we take our control samples, we run each of them at multiple dilutions, and we see which dilution gives us signals that are in that range from 25,000 down to 5,000 or so. And you see the examples of that data in the right plot. And you can see, for example, with sample 3, the signals go down with increasing dilution as expected. And the reason there are differences in signals between the samples is because each sample is different. Each sample has a different amount of CEA in it. But in each case, the signal goes down with increasing dilution. So S3 is the most concentrated sample, and at just the 20x dilution, or the most concentrated, the signal is out of the linear response range. So we wouldn't be able to really get a good value at 20x dilution for that sample, although we could probably for the S2. So we're looking for a dilution that gives us the best response in the range that we want. So we see that at 20x, one sample is out of range high. At 200x, one sample is out of range low. That was the S2. So we really need to pick something depending on where we need to optimize. Do we need to optimize for best sensitivity, if we're not worried about the upper range, or do we need to optimize for a best ability to quantitate in the mid-range or the high range? So that decision may depend on where your threshold is. So for example, this is a clinical assay where your threshold for decision-making is down in the low nanogram per milliliter range, then you would use a low dilution factor. So those are some considerations for optimizing the dilution factor in the assays. I'll just talk briefly now about considerations for running the samples, just some basic experimental principles. This is not exhaustive, but just some things to think about that we put into practice routinely. Each sample should be run in 3 replicate wells or arrays. You really need completely independent wells or arrays to really get a view for the reproducibility, the precision of the assay, to tell you if it's working well or not. With each sample set, you want to run the calibration curve, and I'll tell you in the next slide about how to use that. And then the control samples with each sample set. So notes here, you want to use the same calibrators and controls in every experiment so that you can compare between experiments. One consideration for the samples and the antibodies for long-term use is it's valuable to have fine or small aliquots. We use a fresh aliquot every time. And of course, if you're doing clinical sample sets, remember to randomize everything to minimize the introduction of bias. And I'll tell you how to use this information now in the next slide, some considerations for analyzing the data. So you want to first check to see, check the curve to make sure your assay is working well with a smooth sigmoidal shape, and you want to determine the range where you're getting a linear response. So what is the limit high and the limit low, and then you compare your samples to the calibration curve, noting those that are out of range high or out of range low, and the ones that are in the response range, those can be converted to concentrations through solving for an equation for the curve and then converting signal to concentration. And then it's valuable to calculate the CVs between the triplicates using the values from the concentrations after conversion from signal to concentration. We often will rerun samples that have very high CVs. We'll first visually inspect to see if there was a problem with those samples and rerun them. That's noted here. And your definition of what a high CV is depends really upon the experimental requirements and the assay. And confirm that the control samples, so you run the control samples with every set, those are maybe the same 10 or 15 samples in every set, could be 5, that have a range of concentrations of your analyte, and you want to make sure that the values that you get for those control samples are the same every time with every experiment set. So that would be the next step, is to make sure they have good CVs and correlate well with previous data. So those are the main considerations. If all looks good, then you can interpret the data from your experimental samples. And that would be according to the experimental design and the purpose of the experiment. If it's a case control experiment, doing the statistical analyses according to differences between cases and controls, so forth. These considerations here hopefully get you to the point where you are able to get reliable, interpretable data. And those are the main things I wanted to cover. I will be happy to address any questions after the other speakers present. I'd like to just acknowledge my lab, who really put together all of this work and has done a great job, especially my lab manager, Katie Partika, and a postdoc, Jung Kao, another postdoc, Yuan Tang, and the key collaborators who supplied samples and technology for what I presented here, and then NCI for funding, and my institute, the Andel Research Institute, has supported us. Just briefly, I'll leave this here for selected further reading. You can refer back to some of the key publications on these topics from our lab. And I'd like to give a brief introduction on how bead-based multiplex protein detection works. Dr. Carreno, after me, will talk about applications using this technology. I'm going to give an introduction into how it works. The multiplexing, as I mentioned before, works by having beads with different colors or spectral addresses, and each of those beads can be conjugated with a different capture reagent. So each color bead would get a different capture reagent by which the different capture reagents can be distinguished, and these are all in suspension, which has some advantages for the kinetics of the antibody protein assays. And after the beads then are mixed together to different color beads, the sample is added to the beads, and the analytes are captured to the antibodies attached to the beads. And once that happens, then there's another antibody that is targeting the captured protein. So this is a standard sandwich assay format, and that antibody is tagged with a fluorescent reporter tag. And then the mix of the microspheres are passed through a fluidics detection chamber in single file, similar to fluorescence-activated cell sorting, FACS. And the FACS machine detects two colors. One is the color of the bead, telling which analyte it is, and the second color is the amount of reporter tag, which tells how much of that protein is bound by that bead. So in that way, the assay gives multiplexed information on multiple different proteins and the concentrations of multiple different proteins. And with that, I'll end, and I'll leave it to Dr. Carreno to tell you more about applications using this technology.
All right. Thank you, Brian. We now go to Dr. Beatriz Carreno, who will describe applications of multiplex technology in cancer research.
Thank you, Gwen. Thank you also to Current Protocols and EMD Millipore for the invitation to speak. And what I'd like to do today is go over studies that we have in our lab using a dendritic cell-based vaccine to treat patients with metastatic melanoma. And I'll share with you some of the results of that study, and I also share with you how we have used a multiplex platform to characterize the vaccine and to understand the T-cell responses that that vaccine induces. So as a matter of introduction, I'll briefly describe what dendritic cells are. These are cells that are rare. They're immune cells, and they have a very important function for initiating immune responses. The cells are usually in an immature state, and by sensing signals from pathogens, bacteria or viruses, or also from other cells, these cells undergo a maturation process, and the cells are able to migrate usually from the periphery. They go to lymph nodes and take antigen, and in the lymph nodes, then they act to activate T cells. The cells in the process of maturation acquire the capacity of making cytokines, and they also express in their surface immune activating receptors. And both the cytokines and the immune activating receptors are required for the generation and proper activation of T cells. For this reason, it has been postulated that dendritic cells should be important components of a vaccine, and multiple cellular therapies incorporating these cells have been studied to date. Unfortunately, most of the 60 trials that have been done to date with dendritic cells have had very disappointing results. There have been less than 10% overall clinical responses, and it has been proposed that part of the reason these clinical trials have not yielded the expected clinical outcomes is because the cells that have been used are immature dendritic cells. That means that the cells do not make cytokines. So based on these postulates, we initiated a clinical trial in 2006 where we planned to use a different kind of dendritic cell, what we call a mature dendritic cell. And part of the supporting data to move forward with these cells was that there was preclinical data, primarily from Matthew Measure, showing that there is a cytokine that dendritic cells make called IL-12, which is critical for the generation and expansion of T cells, particularly of T cells of the CD8 type. So we propose as a hypothesis for our study that immunization with functionally mature IL-12 producing dendritic cells will correlate with generating an immunity to the tumor, to melanoma, and that this will improve the clinical outcome. And I'm just going to briefly walk you here through how we generated this vaccine. So we basically take peripheral blood mononuclear cells from the patient. We isolate dendritic cells So it's a personalized vaccine. Every patient gets dendritic cells that are custom-made to themselves. We use a combination of IL-4 and GM-CSF to generate the dendritic cells. Then these cells are mature using, in our case, a combination of CD40 ligand and interferon gamma. And this is based on previous results, which has demonstrated that you need two signals to generate an IL-12 producing dendritic cells. Once these cells are mature, then they're loaded with peptides. In our case, the peptides were from a protein that is expressed in melanoma called GP100. And then these cells loaded and mature are given back to the patient through an infusion. So we treated 7 patients. This is a phase 1 proof of principle clinical study. There were 7 patients that were treated. All the patients were stage 4, which means that they have metastatic disease. And as you can see here, they had multiple lesions that were in visceral organs, such as the lung, the liver. There was a primary objective, which was the immunological response. So this is we were interested in looking to see if an immune response to GP100 was generated. And the way we measure this was by weekly drawing blood from the patients and doing immunomonitoring using a tetramer assay. The secondary objective was to measure the clinical response, and this was done by RECIST criteria. So the clinical outcome of the study, and I'm showing you at the top radiological results from the first patient. The patient had this is indicated here as P1. This patient had multiple lesions when started being treated with the vaccine, and it had a complete response, and this patient remains in remission 5 years after being out of treatment. There were 2 other clinical responses in the study, P5 and P6. These were partial responses, so there was no more than 30% reduction in tumor burden. Of these patients, patient 5 remains alive, and it has a small nodule, but is basically healthy. The point that we wanted to make here is we were interested then in understanding why not all the patients had had a clinical response. And then we turned to characterizing the vaccine and seeing if there were differences between the vaccines that the patients receive. And one of the things we did was we look at IL-12 production by the vaccine. So the vaccine is made in vitro. The cells are mature, and then the supernatants are collected to look at IL-12 production before the cells are given back to the patient. And when we did this analysis, which is a Cox regression analysis, we found out that the 3 patients that had the clinical response, so the clinical response here is represented as time to progression. You can see that the 3 patients that had the clinical response were the 3 patients that produced the highest amount of IL-12, while the 4 patients that had no clinical benefit from the vaccine produced lower amounts of IL-12. So this then brought the question as to why there was a difference in IL-12 production between the clinical responders and the non-responders, and whether in the case of the non-responders, it was just a reflection of a generalized immune defect, or it was something just specific to production of IL-12 by the dendritic cells. So we were interested in investigating whether the dendritic cells had a general immune defect or it was specific for IL-12. So we turned to the multiplex assays and look at the supernatants that were made from the dendritic cells once they were activated. and examine 24, I'm showing you here 24 out of the 41 cytokines that we tested, and we asked whether production of other cytokines was also affected in the non-clinical responders. And what you see here is that, for example, if you look at IL-6 or IL-8 or TNF-alpha, production of the cytokines is seen in all the patients. In contrast, when you look at IL-12 P70, only the patients that were clinical responders, that is P1, P5, and P6, were able to make IL-12 P70. So these data then said to us that the defect was specific to IL-12. It was probably pathway specific also. So then we wanted to look further into what of the two components of IL-12 was being defective in the patients that had no clinical response. So just briefly, IL-12 is called IL-12 P70 because it's composed of two chains. It's a heterodimer made of P40 and P35. P40 can be secreted as a monomer. P35 cannot come out of the endoplasmic reticulum unless P40 is present. So P35 is usually accepted that when you measure P70, you're measuring P35. When you're measuring P40, you're measuring P40, the monomer. So we wanted to see which of the two chains was defective in the patients that had no clinical response. And what you see here, I am showing you the production of P40 or P70 by immature dendritic cells indicated here as IDC, or mature dendritic cells. And what you see is that there is no defect in the patients in production of P40. However, when you look at P70, which is a reflection of P35, you can see that the production is much lower and that the median and the 25 and 75 percentile is much broader, meaning then that there is a reduced induction of P35 in some of the patients. So it looks from this data then that the defect is at the level of P35 production. So so we went and wanted to look even deeper inside the cell as to what might be defective, whether it is interferon gamma or signaling or CD40 signaling. And I'm just showing you here a scheme of how these two receptor and cytokine signal into the cell. Interferon gamma does it through the interferon gamma receptor through the JAK1 STAT pathway, and it's very important in induction of P35. CD40 ligand does it through CD40, a TNF family member, and it does it inside the cell through the TRAP6 P38 MAP kinase. So we took dendritic cells and activated with interferon gamma and CD40 ligand, made lysates, and then we look using the MILLIPLEX MAP signaling at the STAT1 and P35 kinases, their phosphorylation stat upon activation. And what you see here is where it says IDC, that's the baseline, that when you activate STAT1 is phosphorylated by 5 minutes, and it goes up, and it peaks at about 15, and then it comes down. Well, P38, it's a little delayed, and it peaks about 60 minutes, and then it remains up. So based on this information, then we decided that we were going to go back and look at dendritic cells from our patients and see if we could see a difference between non-responders and responders. So I'm showing you here the data for patient 1, which is a clinical responder, and patient 3, which is a clinical non-responder, looking at total stat and phosphostat in two ways. One is with a MILLIPLEX MAX, and one is with a phosphoflow. And what you can see is that the two patients look identical. So based on this data, it looks like there is no defect in the membrane proximal signaling in neither of the patients, and that the defect in P35 induction is not really at the membrane proximal site. So at this point in time, we are looking down the pathway. We're investigating, looking at interferon responsive elements, but we don't know at this point what the defect is. So to conclude, I just want to go over and look at the T cells, our results with the T cells. So I have told you so far that there is a difference in the IL-12 that is made by the dendritic cells from the clinical responders and the non-responders. And now we wanted to see if there was a difference in the T cell responses that were generated. All the patients had an immune response, so it was not the quantity of the immune response, but possibly the quality of the immune response. So we took T cells from, I'm showing you here, 6 of the 7 patients. And what you see is that when you look at the supernatants, when these cells were activated, and you look at the right hand of the panel, you see again that there is a difference between clinical responders and non-responders. The clinical responders tend to make interferon gamma, and that's patient 1, 5, and 6. The clinical non-responders tends to make IL-5 and IL-13. That's patient 2, 4, and 7. So T cells from the clinical responders make more interferon gamma. From the non-responders make more IL-5 and 13. And when you do a ratio, it's significant, the difference between clinical responders and non-responders. So the clinical responders have a TC1 bias response. The clinical non-responders have a TC2 response. And then in summary and as a conclusion, it looks from our studies that IL-12 P70 is important for the priming, at least in metastatic melanoma. That leads to the generation of a TC1 response, and that TC1 response turns into a clinical response. In the absence of IL-12, you get more of a TC2 response, and that leads to rapid disease progression. Thank you very much.
Thank you so much, Beatriz. And we now go to Dr. Deb MacIvor, who will briefly describe the breadth of the MILLIPLEX MAP cancer research portfolio.
Thank you, Gwen. And I also want to thank Brian and Beatriz for their very informative and insightful talks. Brian did a great job of discussing the potential advantages of array and bead-based multiplexing, and Beatriz did a great job demonstrating how she used our bead-based MILLIPLEX kits to enable her to get the results that she did in her melanoma study. So I would like to spend a few minutes telling about our multiplex kits. The first slide that we have here is a comparison of our MILLIPLEX panels using the Luminex xMAP technology with traditional methods such as the ELISA and Western blotting. So if you consider the number of plates or gels required and the total hands-on time that is required to study 10 proteins in 40 samples and looking in duplicate samples, you will see that multiplexing can give you the highest number of quantitative results in the shortest amount of time and in the smallest sample size. In addition, multiplexing has a greater dynamic range and with similar lower limits of detection that's similar to ELISA's. The next slide illustrates many of the panels and kits that are available to cancer researchers. In the center here, We have the 10 hallmarks of cancer, although very recently this has been updated now to 12. And around the hallmarks of cancer, we have the different panels, multiplex panels that can be used to study each of these hallmarks. So in addition to our circulating cancer biomarker panels that we have, for example, we have angiogenesis and metastasis kits. We have cytokines, soluble cytokine receptors, and Th17 and CD8 panels. We have metabolism and endocrine panels, as well as bone metabolism panels. We have intracellular kits, such as those that Beatriz used, and others such as apoptosis. We have NF-kappa B, TGF-beta, RTKs, and others. And we also have cellular metabolism kits to study things like OXPHOS, oxidative stress, glycolysis, etc. Finally, what I'd like to leave you with, I want to introduce a contest that we've just initiated. We're calling it the Multiplex Grant Program, and are asking you to tell us how you would use our MILLIPLEX kits in your research. You will then qualify to win a MagPix, the newest member of the line of Luminex instruments, which is a $30,000 value. So to qualify and for complete details, please visit our website, EMD or MerckMillipore.com/MPX_grant. So thank you all, and thank you for your participation.
All right. Thank you, Brian, Beatriz, and Deb. And it's now time for our question and answer session. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. Let's go ahead and see what questions have come in so far. Brian, I believe this one would be for you. It says, what do I do if I don't have a good standard for the calibration curve?
Okay, good question. Can you hear me fine?
I hear you just fine.
Good. Okay. Just wanted to be sure before I started in on the answer.
Sure.
That's a good question, and one that can be faced regularly if you are targeting something that's more of an unusual protein. You might maybe have an antibody against something you synthesized a peptide for, but you don't have a good protein standard. And sometimes it takes a lot to get high quantities of purified proteins. It can be an expensive process. And in that case, we try to find a cell line or a clinical sample that contains that protein, and then another cell line or clinical sample that does not. You can verify that maybe through Western blot, for example, or gene expression. So you find a sample that contains the protein target of interest and one that does not, and those would be your positive and negative controls. And you can use those positive and negative controls to verify the specificity of the assay. So you should see a nice good signal and a nice clean curve using dilutions of your positive sample, and you should see no signal from your negative sample. So once you've verified that you're getting good, clean, specific signal, then you can actually then use that sample as your protein standard. It's not ideal because you don't know the actual concentration, but you know relative concentration between the dilutions, and it's good enough to set the linear range and the dilution factors according to the methods I showed.
All right. The next question, I believe, Brian, this might be for you as well. It says, what is an acceptable coefficient of variation, or a CV, between repeat experiments?
Okay, I just briefly mentioned that in the section on analysis, where I mentioned that it's a good idea to look at the CV of each sample, and then samples that are above some sort of threshold, you want to look at those more closely to see what went wrong. If you can't identify anything that went wrong, or even if you can, then maybe to repeat that sample, repeat all the samples. And so the question is, what's the unacceptably high CV, and it sort of depends upon the precision of your particular assay. We've found that when everything is working well, we can get CVs around 20 to 30% routinely, and it also depends on if this is discovery research or you're doing more validation of particular markers or something. Then you want tighter CVs. So the CV is the standard deviation divided by the average between the repeats. And so if you see a 20 to 30% CV, you can rely upon that as a reliable measurement. So anything higher than that, you want to take a more careful look at. It doesn't mean that you can't use the data necessarily, but you probably could do better to tighten that up. So that's the range we look at usually.
All right. This next one I think would be for Dr. MacIvor. The question is, Dr. Haab mentioned the considerations necessary for collecting and analyzing data for each sample. How do the multiplex kits address these considerations?
Okay. Thank you for the question. So our goal with our kits basically is to provide our customers with kits that perform consistently from lot to lot. So we do that in several ways. We do include standards, and our standards are always, the new lot of standards is always compared to a reference lot. Our detection antibodies are in cocktails, and they're tested for cross-reactivity within the panel. They're also optimized for both expression and for consistent performance. Our kits also include QCs, so that you can use those QCs to know that your assay is running correctly. And we've also worked out a lot of the dilution factors for the kits, especially in whatever sample type that is appropriate, whether it's serum or plasma or urine or CSF or whatever. And then the Luminex machines themselves actually contain a software to both acquire and analyze data, such as standard curves, the CVs, and sensitivity. And then we offer an analysis software package that actually allows a lot of flexibility in how you design your plate, and gives a lot of different kinds of various reporting tools, such as heat maps. So that's kind of how we address some of the issues that I know Brian experiences doing some of the home brews and things like that.
All right. The next question, I believe, is back for Brian. It says, what's the best way to convert from fluorescence to concentration?
Okay, right. If you are doing, as Dr. MacIvor mentioned, homebrew assays, you're developing your own prototypes of assays, you might not have some built-in software, as just was described, for doing that, and so you might need to just get that going in your lab. I think with the Luminex platform, it's taken care of, but you might have something where you need to do that by hand. So that refers to taking the calibration curve. So I mentioned you have a curve of a standard, and you compare that to your unknown samples in order to convert fluorescence to concentration. So there are Excel tools for doing that, and it's the solver packages or functions within Excel where you can take the curve and you can solve to a 3-parameter fit, and then you can plug in the Y value, which would be the fluorescence, to solve for concentration. That's for doing many samples, that's not a great way to go. It's just very time-consuming, requires a lot of manual effort. We've found a couple of software packages that work pretty well, very flexible for entering your calibration curve, finding the linear range, taking a whole set of unknown data, your sample data, and then converting them to fluorescence. One is from Hitachi. I think it's called, it's a multiplex analysis tool from Hitachi Software, and that's the one we use. I'd recommend that. There's a few other software tools out there for that purpose.
Okay, and we have time for just one more question, and I found one here that seems to be really pretty generally but important. I believe this would be for Brian. It says, what is the best way to choose your control samples?
Okay, so these are the samples that are used, I mentioned, in each experiment set. So whenever you do an experiment on a different day, you want to have a small set of samples that you run every time, the same samples. And the purpose of that is to make sure that you're getting reproducible results from day to day. You're not using these samples to calibrate your signal or to do any adjustments or anything like that, but really you treat them the same as you treat all your unknown samples, but you know what you should get because you've run them before. And so this is a set of samples that you have a lot of material available for, and that you know cover a range of concentrations for each of your analytes, so that you can easily see whether you're getting a good correlation in results. So if they all are low or all are very high, then that's not a good set of control samples. So you want some samples that are high, some that ideally are medium, and low or negative. And then that should give you a good way to determine whether the assay is performing consistently from day to day.
All right. Well, we are just about out of time, so I have to conclude the Q&A session. I want to let everyone know that today's webinar has been recorded and will be available for viewing in the next few days. We'll send each of you an email with details of how to access the recorded webinar, along with a PDF of the slides and instructions on how to customize and print a certificate of attendance. You can also access the certificate of attendance right now from the Resources tab at the bottom of your screen. In addition, as a token of our appreciation, you can download the latest technical poster of Dr. Carreno's research study titled Characterization of Protein Biomarkers of Metastasis in Melanoma, courtesy of our sponsor, EMD Millipore. So this concludes today's webinar. We hope it was both informative and educational for you. On behalf of our speakers and from me, Gwen Taylor, and our sponsor, EMD Millipore, we look forward to your attendance at future events from Current Protocols. Thank you so much for attending.